Let's give a big Texoma Cattlemen's Conference welcome to Dr. Mark Thallman.
Thank you. Well, it's a privilege to be here today, and appreciate the opportunity to visit with you about some of the things that have changed in the cattle industry and that we think may change in the future. I'm going to start with breed differences in exotic breeds. This is fairly near and dear to me. This is the Germplasm Evaluation Project that's a flagship project at the Meat Animal Research Center. And we've evaluated 37 breeds from about 1970 to the mid-2000s, and then over the last 10 years we've focused on evaluating the 18 breeds that are having the most influence on the beef industry. We've also done a lot of work on crossbreeding, and all of you know that that's been one of the real changes in the beef industry since the 1960s. A lot of advantages to crossbreeding. There's been quite a bit of change in terms of developing new breeds and composites in the beef industry in the last 50 years. Especially here in Oklahoma and Texas, you're familiar with a lot of the American breeds that were developed in this part of the country. You all know that visual appraisals had a big impact on genetic improvement in the beef industry. You know, I think most of that's good. Sometimes, as indicated by this bull towards the bottom, maybe we get the idea that we want something and we go a little bit too far with it. Performance recording, it has been a big change in our beef industry over the years. And the Beef Improvement Federation has driven a lot of that change. Our annual convention will be in Manhattan, Kansas this June, and if you're interested in performance recording, it'd be a great opportunity to come to the BIF convention. Genetic evaluation is something that I think is probably transformed genetic improvement as much as anything that we've done in beef cattle. That's the submission of records into a national database and computing expected progeny differences, or EPDs, from that consolidated data. And as this slide indicates, we've seen really strong trends for genetic improvement as a consequence of that for the traits that we've had EPDs for. And results basically indicate that for the traits we haven't had EPDs for, we make a lot less progress. My colleagues at the Meat Animal Research Center and I calculate a set of crossbreed EPD adjustment factors each year to help put EPDs on a constant base for various breeds that are evaluated in the United States. I hope you're aware, without adjusting the breeds to a common base, they're not directly comparable. So that's something that has been going on for a bit over 20 years now. And then one of the things that's happened more recently, really over the last 6 years or so, is the incorporation of genomics into our EPD system. And a lot of you have probably heard about genomics for 20, 25 years. You know, probably if you'd come to this conference and we were talking about future technologies, anytime up until about 5 years ago, genomics would have been one of the things that was talked about a lot that hadn't really had any impact yet. But since about 2010, genomics has been incorporated into our genetic evaluation system, and first in Angus in 2010, but we've got over half of those top 18 breeds that now have genetic evaluations that make use of genomics in one way or another. And that's really having an impact, and it's allowing us to have EPDs that are more accurate at a younger age. And that's really relevant for those of you who buy bulls to go out on commercial programs. That the accuracy of those young bulls is better now with genomics than it was before. Well, I think most of you are familiar with using genomics for parentage determination. We can use it we've been using it to verify parentage for a long time, but we're now seeing a lot of people use multiple sire natural service breeding pastures and then determine the parentage from that. And that can be beneficial both in a seed stock operation or commercial production. And I think most of you are familiar with using genomics to test for genetic defects, and you're probably aware that there have been a lot of genetic defects that kind of gained general attention in the last 10 years, and a lot of those have been now controlled through the application of genomic testing. I'm involved in developing an approach to low-cost genotyping and sequencing technology in collaboration with commercial partners through a cooperative research and development project. And one of the real limitations that we've had in the adoption of genomics technology is just the cost of doing the testing. And so we're trying to drive that cost down so that genomics can have a lot more impact on the industry than it has so far. And our hope would be to drive that down to the point that genomics is profitable to use in commercial production as well as seed stock. At the Meat Animal Research Center, our primary focus on genomics is to move beyond the within-breed prediction of using genomics that has already contributed a lot to the beef industry and move beyond that into multi-breed genomic prediction and prediction for traits that are not routinely evaluated through the EPD system. So those traits that are really expensive to measure and are very limited, we evaluate our animals pretty extensively for those traits, and our goal is to develop genomic tests that help you to select for those traits. That's a more challenging goal than doing within breed evaluation for traits that are routinely recorded. So it's going to still take a while to get there, but we think it's worth pursuing. A moment ago, I mentioned marker-assisted management. This is the idea of doing genomic testing on commercial cattle to allow commercial producers to make decisions based on genomics. And it's not so much about improving the genetic merit of the animals, but changing how you manage those animals depending on what their genetic potential is. And there have been some entries into this market. Cargill actually put a lot into marker-assisted management in feedlot cattle several years ago. Cactus has put a lot into it, and currently there are a couple of products out on the market, the GeneMax product from Pfizer or Zoetis and the American Angus Association, and Igenity has products along those lines as well. So I think eventually we will see the same kind of technology that we use to evaluate genetic merit through EPDs applied to commercial cattle. But in that case, the objective is not to evaluate the genetic merit of the animals, but evaluate the phenotypic potential of those animals so that you can manage them accordingly. And it could be either in terms of determining the value of the cattle, so you determine which ones you want to buy and how much they're worth to you, or it could be how are you going to use technologies like implants or beta agonists, etc. So there are a few examples of early entrants into this market, and that would be verified beef and top dollar Angus, and maybe overlooking some others. But I think this is something we'll see eventually. It requires there's some fairly major hurdles to restructuring the marketing system for the beef industry for this to really catch hold and do what it could. Introgression is a term that geneticists use that means that if we had a form of a gene in one breed and we wanted to introduce that gene into another breed, we would cross those two breeds and then repeatedly back cross to the breed that we want to introgress into, but always selecting for the gene that we want to move into that breed. And there's a variety of examples where that's already happened, and this particular bull would illustrate those. That's moving black and polled into the Simmental breed, and also into a lot of other breeds as well. But there's a lot of other opportunities that are going to come to do that, and with better understanding of genomics and better understanding of individual genes that have big effects on traits that are of economic value, there's going to continue to be incentives to move new little pieces of the genome into breeds that they weren't in before. And genomics makes that work a lot more efficiently than was possible without genomics. Now, you probably know that Simmental had already become fairly black and fairly polled before we had DNA tests for those traits. But those traits accelerate the ability to make Simmental black and polled. And some of the other breeds that have come along that have become black and polled since then really benefited from DNA tests for those traits. And, I think eventually we're going to see opportunities to move other things into a lot of breeds that they weren't in before. And this is an issue that some of our breed associations are going to have to grapple with, I think, because a lot of our breed associations are open to this. They have registry policies that allow grading up to their breed from other breeds. But a couple of the big breed associations and the oldest breed associations in the U.S. have closed herd books. And sooner or later, that's going to put them at a real disadvantage unless they open those herd books up to allow introgression. So just something to be thinking about out there on the horizon. One example that I'll give you for that is moving the inactive form of myostatin into other breeds of cattle. The inactive form of myostatin is something that you might know of. You might think of as a genetic defect, or you might think of it as a real advantage. It's what we call double muscling. And I know there's a lot of variation of opinions on double muscling, but it has some pretty profound effects on beef production. It really changes body composition. It really increases muscle, decreases fat, and it decreases marbling, but it has a tremendous effect on improving tenderness. And not just improving tenderness in the middle meats where we get our steaks from currently, but it actually improves tenderness more in the end meats, so in the round and chuck. And so it has the opportunity to extend the amount of the carcass that we could sell as premium cuts going into steaks. So it's something that the industry ought to be thinking about. And, it's present currently in Belgian Blue and Piedmontese, and there's minor frequencies of it in some other breeds. But it's the kind of thing that we ought to be thinking about. The challenge with introgressing double muscling into other breeds and taking advantage of it is that we really have to have a different marketing system than what we have today. You don't want to just introgress that and then sell into the current commodity marketing system that we have. But for branded products or a change in marketing system, it could have a really profound effect. It's probably the single one gene that the beef industry could benefit from the most that it's ignored. There's another way to change cattle besides introgression, and that's what we refer to as transgenics. And I think you're all familiar with transgenics in some form. In the plant world, it's called GMOs, genetically modified organisms, and they get a bad rap, as most of you know. And there's challenges about introducing a gene from one species into a totally different species in plants, and there's been a lot of pushback against that. Those challenges are a lot greater when we move to animals, and but just recently we've had the first transgenic animal product approved for use in commercial production, and that was a gene introduced into Atlantic salmon that FDA finally approved after about a 20-year battle to get that approved for commercial production. So the challenges and the drawbacks to using transgenic technology, particularly bringing in moving genes from one species to another, are very substantial. And I think because of that, we really haven't, I'd say for the most part, seriously considered doing that in livestock production. There have been transgenic cattle produced for over 20 years, but it's mainly been as a research endeavor, and I think that both the industry and the academic community have pretty much coalesced around the idea that we just didn't want to fight the public perception battles that would exist with that. And the other thing that I should point out to you is it's more severe when you bring a gene from another species into a mammal than it is when you do that in a plant. Plants can tolerate that kind of stuff really well. The genome of mammals is a really finely tuned system. And generally, when we have brought something like a different growth hormone gene into livestock, we've seen a lot of adverse effects from it. And so I'm bringing this up just because we want to talk about what's out there. I'm not suggesting that we ought to be doing what we'd think of as traditional transgenics. I've generally been of the opinion we should shy away from that, and I think we're pretty well agreed within the livestock community that that's what we want to do. But I want to bring up with you a new term called gene editing. And this is a form of, they really are transgenics, okay, but they can be transgenics of a different type. So it's a different way of producing transgenics. And as this slide is trying to illustrate, it's making a very targeted substitution in a very specific region of the genome. And in principle, this could be done in a lot of different areas of the genome simultaneously, and it can be done a lot more efficiently than the old forms of transgenics. And one of the interesting things about gene editing is that most of the applications that are being proposed for gene editing, in livestock at least, are to make substitutions that naturally occur within cattle, but to just do it faster. So you could think of it as it's like doing introgression, but just doing it in one generation rather than having to take 4 or 5 generations to do it. And so there is a company that is offering this as a commercial service. I think they're doing maybe a little bit more work in swine than in cattle, but these 2 polled Holsteins on here were produced through this technology, and I've never really understood why you couldn't introgress polled into Holstein pretty easily, but apparently there's enough of a penalty and genetic merit from doing that Holstein breeders have been pretty resistant to do it. And so the folks marketing this technology saw that as being a great demonstration project in cattle. But there are a lot of other things that could be done with it. And, to some extent, a person could do a genomic test on an animal that's had one of these substitutions through gene editing, and you wouldn't really be able to distinguish, unless you went back and traced through the pedigree, you wouldn't be able to distinguish, did that happen through conventional breeding with introgression, or did it happen through gene editing? And so the consequence of that is that proponents of this technology say this isn't transgenics, we're different than transgenics, and this is okay, and the public is going to accept it as okay. And, so there's a lot of debate about, well, can we get by with that, or are we not going to get by with that? So I'm not on either side of that issue, but I think you need to be aware of gene editing. This is one that you are going to hear about, within the next 5 years. And, as an industry, we're probably better off to say, yeah, this is different. Let's try to take advantage of it if we can. But I'm sure there will be activist groups that will be labeling this as, developing frankenfoods and all the other things that we've heard before. I mentioned traditional breed formation before. And in this slide, what I want to try to represent is we've got a lot of composite breeds, and you all should be familiar with a lot of them in this part of the country. A lot of them that are Brahmin crossed on some sort of Bos Taurus. So we've got Brangus, Sembra, Brayford, Santa Gertrudis, Beefmaster, and several others that have been developed over the years. And these breeds have been developed basically on the basis of breed percentage. And so the idea is once we've formed the breed and gone through several generations, we've got a group of animals that are either 3H Brahmin or half Brahmin, and then the rest is whatever the other Bos Taurus source of germplasm was. And so on average, they will be that percentage. After several generations of selection, that percentage probably varies a little bit, and there'll be some selection for either the Brahmin contribution or the Bos Taurus contribution in various locations in the genome. In the case of Brangus, obviously we're selecting pretty hard for the regions where Angus makes them black and polled. There'll be other regions where the Brahmin contribution is under favorable selection because it makes them more adapted to our environment in this part of the country. But I think as we go forward, we could look at breed formation from a different perspective, leaning on what we learned from genomics. And in these composites especially between Bos indicus and Bos taurus, at any particular location in the genome, we ought to be able to assess, here is the Brahmin more favorable or is the Bos taurus more favorable? And then let's put directional selection on that region of the genome. And then at another location in the genome, the other source is going to be more favorable. And applying that kind of selection over a period of several generations using genomics could get us to a place where we have composite breeds that now are fairly uniform in terms of their source of origin at any one location in the genome, but they're going to be more uniform as a breed, and they're going to be more adapted to the environment that they need to be produced in, and they're going to be more productive as a consequence of that. And so this kind of approach could be applied to the existing breeds that are already out there, or it could be applied to forming new breeds. So I think that's something, it's an area or an opportunity with genomics that we really have not taken advantage of yet. we're just kind of now approaching the point where we can estimate what the breed effects are at positions in the genome in order to enable this kind of selection. But I sure hope that within the next 5 years we'll be seeing this utilized. There also are reciprocal breed effects that are important in terms of how we build a composite breed, especially composites between Brahmin and Bos Taurus. And some of those are traditional maternal effects, but some of them are not. And so it does actually matter how you combine, whether you use a Brahmin bull on a Bos Taurus cow or a Bos Taurus bull on a Brahmin cow. And the results that you get will be very different for a lot of different traits depending on how you go about that. Phenomics is a term that I want to introduce to you. Phenomics is kind of saying measuring lots of phenotypes or different kinds of data points on cattle and doing it on a large-scale basis across lots of animals and lots of different measures. And so the idea would be that we want animals that we've extensively evaluated for economically relevant traits, but then we might we'd also collect a lot of what I'd refer to as physiological indicator traits. So these could be things like gene expression, metabolomics, proteomics. The approach is to break complex traits down into simpler component traits that are more likely to be affected by one gene or a small number of genes, so that we can do a better job of predicting those components and then add it all back together so that we meet our objective of predicting the economically important traits. So it's probably not something that you all would be particularly getting involved in, but it's something that has a lot of potential to provide you with tools that allow you to both breed cattle more effectively and produce and manage cattle more effectively. Don't know how many of you have heard the term metagenomics before. Metagenomics is the idea that DNA sequencing has become so inexpensive now that we can go out and collect a sample of biological material from any part of an animal or from the environment, from the soil, from water, from just about anywhere, extract the DNA from that sample and find out what microorganisms are present in that sample. And 10 years ago, we had no idea what the spectrum of microorganisms that exist on Earth was. We still are just scratching the surface at being able to understand what that is. But each one of you has probably 10 times as many microbial cells in your body as you have mammalian cells. And for a ruminant, it's even a higher proportion. We have microbes all over our skin, and the populations that's on the skin on one part of your body is totally different from the population of microbes that's on the skin on another part of your body. And then that's going to be very different from what's inside your gut, and it's going to vary very dramatically from one part of your gut to another. And you all know how important microbial populations are in the digestive system of ruminants. But there's all kinds of microbial communities that colonize all of the respiratory tract. And there's things like Mannheimia that's just there all the time in cattle. And what we know now is that when those cattle get really stressed, some viruses come in and start infecting the reproductive tract, and then those same Mannheimia can be transformed and become a really serious cause of bovine respiratory disease. So from a research perspective, there is an enormous amount of effort going into studying the microbial populations at all of these different levels. And this is another one that, maybe it's not something you get involved in directly, but I'm really confident this is going to have a major impact on what you're doing 10 years from now. And, I would hope that maybe we will eventually learn how to breed microbes that will be more efficient at converting roughage into beef protein, and that will produce less methane than what we're doing right now. One of the things we're learning is there's this class of microorganisms called methanogens that take about 10% of the energy that cattle eat and turn it into methane that they belch out. And this doesn't seem to benefit the cattle, but they're all there doing it. And how much they belch depends a lot on what they're eating. But there's a lot we don't know about it. We have been, I guess, fairly effective in managing that. That's a lot of what ionophores does, is it shifts that population. But I think there's a lot more that could be done as we start to learn more about these microorganisms. I think that there are some opportunities to breed cattle that respond differently to specific pharmaceuticals. I'm going to move into reproductive technologies. You're all familiar with artificial insemination, with the effects of estrus synchronization, automated estrus detection, embryo transfer. We're just starting to see people do genomic testing of embryos prior to transfer of the embryos. That's been a bit of it's been something we've been technically able to do for quite a while, but it's been difficult to make work economically because the tests have been pretty expensive per test, and then if you're going to test a lot of embryos and only transfer a handful of them, then that cost per embryo that you transfer gets really high. So, but I think as we can drive the cost of the testing itself down and make it more, there's also a timing issue that if you're going to sample an embryo, you'd like to know really soon whether to transfer the embryo or not. But I think we'll see this more and more as we go into the future. There is the idea that we can reduce generation interval by harvesting oocytes for in vitro fertilization, not from this calf's mother, but from the calf herself. So harvesting oocytes from prepubertal females could allow us to cut generation interval on the female side from 2 years to a year. That could have a pretty big impact on rate of genetic improvement. I think most of you are familiar with cloning and know it is something that's terribly expensive and has some side effects that aren't real desirable. But there's been quite a bit of press lately on a group in China that is planning to build a factory to develop cloned embryos on a massive scale. And we'll see whether it happens or not, but I personally don't think that's infeasible. I think that improving the efficiency of embryo cloning incrementally to try to use cloning in genetic improvement of cattle is a wasted effort. But I think that there is pretty good plausibility that if we looked at it from an engineering perspective and said, how would you develop a manufacturing process to do it, I think it's actually not so far-fetched as it might sound. So reproductive technologies for just improving commercial production. We've used controlled breeding seasons for a long time. I think personally, I think we've maybe misused them in some ways, but certainly there's been a lot of benefit from that as well. Pregnancy diagnosis is certainly a technology that has improved productivity a great deal. Breeding soundness exams. We have the potential for AI use in commercial herds. We've had some use at a few percentage of the commercial cows in the country for a long time. It hasn't increased a lot. I think it did increase a bit last year when bulls got so expensive. And there certainly were some economic analyses showing that it probably could have increased more than it did. There's opportunities to induce estrus in cows that are postpartum anestrus using some of the synchronization drugs and protocols. And I think we probably could use those more than we do as an industry, particularly on the fraction of your cows that are late calving. So rather than culling cows and turning cows over faster, some of those cows could stay in the herd for quite a long time if we'd just give them a little bit of a boost. I know as a geneticist I'm not supposed to say that, but I'm going to tell you, if you think you're improving the genetic merit of your cows by culling the late calvers and opens, I think you're wasting your time. Maybe if you can keep your herd in the family for the next 3,000 or 4,000 years, your descendants someday will appreciate what you did. But natural selection is something that operates over millions of years, maybe thousands of years, but it's not going to have much effect over your lifetime. So, and I heard just the other day that there's maybe some pretty decent evidence that if we applied estradiol at the right time of the estrus cycle, that actually makes the cow's uterus more receptive to having a fertilized embryo in it and may improve conception rates. So, I think there will be some various kinds of treatments that we'll be able to do in the future that will allow us to improve pregnancy rates and tighten up calving distributions, especially targeting some of these things to the cows that are calving at the end of the calving season. There's some pretty exciting work going on to identify rare recessive defects that result in embryonic mortality. And by selecting against the ones of those that are starting to creep up in frequency within a breed, I think there's pretty good potential to improve fertility. And this will be something that will be mediated through genomic selection at the seed stock level. So pretty easy to implement. Twinning's been around for a long time. Fetal programming is a new hot topic, and it's basically the idea that the nutritional environment that a fetus's dam is exposed to at various times and in various ways throughout pregnancy can have quite a bit of impact on how that fetus is going to grow and develop as either a steer or as a heifer going into the cow herd. And, for example, a cow who gestated in a fairly sparse nutritional environment may think that nutrients are really scarce and they're going to use them in a really stingy way, whereas one that gestated in a very plush environment thinks, well, the nutrients are free and everywhere and kind of burn through them pretty fast. So another really exciting area of research, and I think it's probably going to impact the way that you manage your cattle sometime in the next, within the next decade anyway. Sex semen is a technology that I'm really excited about, and I'm going to explain to you why in a few minutes. So where could we be going in the beef industry? I think where I'd like to see the industry go is more specialization as far as terminal genetics and maternal genetics. A challenge we have is loss of complementarity. All of the breeds are becoming more similar to one another. They all have similar breeding objectives. I'd like to see some divergence. I think the greatest opportunity to improve cow-calf efficiency is to have small maternal line cows bred to growthy, high growth bulls that have good direct calving ease, but lots of growth, lots of carcass merit. And I think that's the most efficient kind of beef production system we can have. The problem is every breed wants to be a general purpose breed. What's changed? Sex semen, I think, could be a real game changer in this. I know there's challenges with using sex semen. I haven't kept up with exactly how well they're addressing those challenges. I'm not here to endorse or not sex semen, but to the extent that sex semen becomes practical for use, the biggest challenge in having maternal lines bred to terminal bulls is replacement rate. And with 50-50 sex ratios, you just have to keep those maternal cows bred back to maternal bulls through 3 or 4 calf crops before you can start switching them over to terminal bulls, just to have enough replacements to keep the program going. Sex semen could reduce that quite a bit, and we could have maternal cows that are being bred to maternal bulls for their, let's say, first 2 calf crops and then switch them over to terminal after that. So I think that an optimal beef industry would look from a genetics perspective, a terminal crossbreeding system with specialized maternal line herds, use sex semen to make that work. Those herds should be selling nearly all their females at 3 to 4 years of age after those females have replaced themselves. My philosophy is that small herds should purchase 3 to 4 year old bred cows that are bred to produce terminal cross calves, and that they should just be buying in these bred cows, use them as long as they can, and then buy another bred cow as a replacement. So why would a small producer calve out heifers? And here I'm talking especially about one bull herds, but probably under 100 cows this may apply to as well. So, part of it is tradition. As an industry, we've basically felt like we should produce our own replacements. That could be a reasonable argument. I'd challenge it, I guess. There's the pride of producing your own replacements so that you can produce continual improvement in your herd. I think that's a valid reason to do it. The most interesting explanation that I've heard for why you would do it is that, it makes a really great topic of conversation at the coffee shop that, oh yeah, I had to go through this Baxter back story to get my heifer in and pull this calf last night at 2 a.m. and I saved the calf and the cow's doing great, and I'm really excited to be in the cattle business, right? So, for people that, if that's important to you, then that's a good reason to be doing it. If that's not what you want to be doing, though, I have a hard time seeing if you value your time and you're keeping back 5 heifers a year, how can you justify going out and spending a lot of time checking those over a period of a month or more? What's the return on that investment of time? So if you're looking at it from that perspective, I think you're a lot better off to just not be messing with heifers. For the small producer, most of the time, I think the heifers are running with the cows. That's suboptimal. In a bigger operation, we almost always separate the heifers out and manage them differently. If you're a small producer, especially the one bull herd, you're having to breed your heifers to the same bull you're breeding your cows to, and there's all kinds of challenges with that. So I think this is something that as an industry we need to really rethink. The challenge is we need a new category of producers that we don't have in large numbers. They're specializing in maternal genetics and that are thinking about, instead of selling a bred heifer, I'd like to see them breed their heifers for about the first 2 calf crops so that they can replace those females before they sell them, and then sell generally a 4-year-old, maybe sometimes a 3-year-old bred female. And I think if we had a market for that, it'd be a lot more efficient system than we have now. And to me, the real game changer on that is the ability to use sex semen. I don't think probably those folks could afford to just use sex semen, but I think integrating it into a system could shift the sex ratio enough to make it work. So I'm going to stop there.
All righty.